Good afternoon, everyone. My name is Juan Ignacio Gonzalez Pacheco, and I'm currently a doctoral fellow at the National Technological University, Mendoza Regional Faculty, Argentina, where I also serve as professor at the Transport Phenomena Chair in the Chemical Engineer career. My thesis and scholarship supervisor, Dr. Mariela Maldonado, who is here with us, and I would like to express our gratitude for the chance to present our research at this conference. Our presence in the stunning city of Singapore wouldn't have been possible without the incredible support from the organizing team. Today, I'm excited to share our findings on quantitative analysis of the diffusion process of a natural dye within a prebiotic sweetened food matrix. Erythrocin, commonly referred to as red number 3, is a synthetic red dye frequently found in a variety of products, including food, cosmetics, and pharmaceuticals. This dye is primarily used to boost the visual appeal of candies, desserts, and beverages by imparting a vibrant red color. The importance of erythrocin lies not just in its widespread application, but also in the growing health concerns surrounding its consumption, particularly its potential links to cancer. The colorant was first discovered in 1875 by Swiss chemist Carl Kussmaul at the University of Basel, nearly 150 years ago. It was soon commercialized by the local company Weisschedler and Bosch for dyeing wool and silk. Its use as a food dye was legalized in the United States with a Pure Food and Drug Act of 1906, which was almost 120 years ago. The FDA approved it for food coloring in 1971, but concerns began to surface around 1990 regarding its potential as a cancer-causing agent. By 2006, reviews were being conducted to explore the connections between erythrocin and certain type of cancer. It wasn't until 2019 that a ban on its use was proposed, and finally on January 15, 2025, it was completely banned. As you can see, it took an astonishing 60 years to put an end to a substance that never should have been approved as a food coloring, given that its original purpose was not related to food at all. Erythrocin is the most widely used colorant in the world, and it's essential to recognize its controversial status. While it has been banned in cosmetics for the past 20 years, it continues to be allowed in food and medicine. This means that although it's not safe for application on the skin, it is deemed acceptable for consumption. This inconsistency is certainly noteworthy. A lot of studies indicate erythrocin may promote tumor growth in animal models. Research shows potential DNA damage associated with erythrocin exposure. Erythrocin's metabolic byproducts have been linked to oxidative stress in cells. The news has gained significant global attention, with widespread discussion surrounding its application. While it's remained banned in the United States, it hasn't yet faced similar restrictions in other parts of the world. This information has been widely known, yet remains unaddressed. The following summarizes the scientific evidence behind what is arguably one of the biggest hoaxes in history. As you see, it not only contributes to cancer, but also disrupts the microbiota, which is the central topic of discussion at this conference. What are the impacts of erythrocin on microbiota? Well, erythrocin may disrupt the balance of gut microbiota, leading to dysbiosis. Studies indicate potential to reduce beneficial bacteria such as lactobacillus. Altered microbial composition can impair gut health and immune function. And concerning the health implications of dysbiosis, this is linked to various health issues, including obesity and metabolic disorders. Changes in microbiota can affect drug metabolism and efficacy, and long-term effects may include increased susceptibility to infections. What options do we have? Are there alternatives available? Well, back in 2017, Dr. Maldonado suggested using different dyes to color cherries, like brilliant blue, which has an acceptable daily intake that is 60 times greater than that of erythricin, the latter between 0 and 0.1 milligram per kilogram. Following this, Justi and Sigurdsson and Justice, introduced the idea of using radish red, a natural dye. Now I'd like to present another natural alternative. We propose gardenia red, which is derived from the Gardenia jasminoides flower. What new challenge does the industry need to address? Well, in order to create a cherry dyeing process utilizing natural colorant, we needed to design the set of experiments. This involved varying the types of colorants, adjusting their concentration in hypertonic prebiotic solutions, and experimenting with different temperatures. We opted for a composition of 50% sucrose and 50% xylitol, which allows for easy scalability of the process. Why do we use prebiotic xylitol syrup? Because it provides a sweet taste and lower calories, prebiotic effect on gut health. Xylitol positively influences gut microbiota composition by reducing harmful bacteria while supporting beneficial strains. This xylitol is a naturally occurring sugar alcohol found in a variety of fruits, including strawberries, plums, and blueberries, as well as in certain vegetables. In industrial production, hemicellulose derived from plant cell walls and birch wood is first broken down through hydrolysis to produce xylose. This xylose is then transformed into xylitol through a catalytic hydrogenation process. Research indicates that xylitol promotes the growth of beneficial bacteria such as bifidobacteria and lactobacilli. Xylitol consumption has been linked to a reduction in pathogenic bacteria, contributing to a healthier microbiome. Clinical studies demonstrate that xylitol can improve gut health parameters, including bowel regularity and microbial diversity. We initially developed 18 formulations using sucrose along with various concentration of polyols, specifically lactitol, maltitol, and xylitol, along with their combinations. We choose formulation F8, which consists of 50% xylitol and 50% sucrose, as I mentioned it previously, because it successfully remained crystal-free for 2 years. Here, I like to talk a little of the mathematical propositions we have developed. We created a mathematical model in order to recreate the movement of these chemical substances, of these colorants, in order to determine if it's technologically possible to replace the artificial colorant by the natural one. To develop a non-dimensional equation governing the variation of A redness with respect to radius and time, we can establish the following dimensionless variables. We have theta here, Here we have theta, which is the non-dimensional time. We have R and E in capital letters, and these are the non-dimensional radii in the epidermis zone and in the center of the cherry respectively. and A is the non-dimensional redness parameter to evaluate the diffusion of pigments inside the fruit's flesh. Here we use 3 boundary conditions. I didn't show it because the slide will be a little complicated, but we had an initial condition and then we have 2 boundary conditions. We have one located at the center and then other at the periphery of the fruit. Here we use a rubbing condition or mixed boundary condition because we try to establish a double diffusion boundary at the flesh-skin interface, trying to explain a simultaneous migration of substances in that interface. Given the particular condition set on the experiment, the only variable to be determined is were the effective diffusivities due to the high mass Biot number. This was achieved by setting this diary to 220 revolutions per minute, reducing the mass convection resistance proportionate for the global fluid movement. Using the non-dimensional A parameter, we calculate the effective diffusivities and the HM values. We employ an iterative nonlinear regression approach. This method utilizes both least square and minimum chi-square methods to ensure precision and reliability in our results. Here are the samples of erythrocin and gardenia red with a measurements variable for dye diffusion, focusing on the redness A parameter. Here we show the kinetics of dye entry and exit, along with the variations in effective diffusivities of erythrocin and gardenia red at the same concentration but under different temperatures. In both cases, as you can see, the patterns are the same. However, major values were obtained in the gardenia red dye. These results indicate that it's possible to technologically replace the artificial colorant with a natural alternative that what is the red gardenia colorant. The surface plots on the right were generated to illustrate not just the kinetics of dye release, but also its variations within the fruit itself. Meanwhile, the graph on the left highlights the temperature concentration synergistic effects on the transfer of dye species. The matrix employed in this test is a porous structure. This allows for the diffusion of polyalcohols, resulting in a sweetened fruit that possesses characteristics akin to a prebiotic syrup. With each impregnation, the complexity of the system expands, which subsequently hinders the diffusion of molecules. This results in an increased tortuosity of the diffusion phenomenon. Tortuosity in foods refer to the complex pathway that fluids must navigate through a porous fluid matrix. This concept is crucial as it influences moisture retention, texture, and the rate of flavor release in foods. Higher tortuosity often leads to slower diffusion rates of substances within the food, impacting overall quality and shelf life. Xylitol, an humectant additive that forms syrup, adheres to the pore walls, delaying the dye's diffusivity. Here, I present the graphical abstract of my latest paper, which I worked on with my thesis supervisor. It encapsulates the entire process of prebiotic cherry coloring and candied preparations. In summary, I can say that approximately 60 years elapsed between the initial approval and the subsequent ban of erythrocin. During this period, this legally sanctioned food additive has been linked to various health issues affecting populations worldwide. It is evident that much of the harm could have been mitigated or avoided entirely had potential conflict of interest in research been addressed appropriately. In light of our commitment to scientific integrity and the responsibility to advance knowledge for the benefit of humanity, we must recognize the availability of new and safe alternatives that are technologically feasible for investigation. This perspective encourages us to shift our focus from the question of what we should eat to sustain our health to a more insightful inquiry into which substances are beneficial and supportive of the healthy development of our microbiome. Given that the microbiome plays a critical role in both physiological and psychological well-being, this realignment is essential. Moreover, it is imperative for the scientific community to prioritize dissemination of knowledge that serves the greater good, not only in developed regions with ample resources, but also in areas where access is limited. Viewing humanity as a holistic system parallels the relationship between the microbiome and the human body, reinforcing our responsibility to contribute positively to public health on a global scale. Thank you very much for your attention.